Hey, alas we came. In this video we are going to discuss about the topic bioinformatics, the rest of the earlier upload. Keep learning. We are always there to help you. Please note one thing. From today we will upload one of our daily videos in favor of 6th semester students. So that Kerala University has declared the date for examination. Support us. Comment your suggestions. Database. Or convenient systems to properly store, search and retrieve any type of data. Share large amounts of data and support large-scale analysis by easy access and will data updation. Biological database. Carries information such as DNA, RNA, protein sequences, 3D images, organism, specific information and information from e-literature. Classified into primary, secondary, derivative, nucleic acid database. Organize and distribute nucleotide sequences from all available sources. Example: EMBL, GenBank, DDBJ, European Molecular Biology Laboratory, EMBL. European Node of 3 Ways International Nucleotide Sequence and Annotation with comprehensive global coverage. Key goal is to integrate nucleotide sequence and annotation into a wealth of bioinformatics resources also offered at the EMBL and elsewhere. Access points to EMBL nucleotide sequence database data include SRS. Homology search tools. The Sequence Version Archive, SVA. FTP site. The Genome's web server, for completed genomes, and sequence retrieval by accession number, DB Fetch. WSDB Fetch and NetServe. It offers a wide range of opportunities to search and download data through the help of access points. GenBank. Most comprehensive and annotated collection of all publicly available DNA sequences. DDBJ. EMBL and NCBI exchange data on daily basis. Important source of data for GenBank. Direct submission from scientists, and the options for submitting data to GenBank include Bankelt, Sequin, Macrosend, TBL2ASN. Keywords used in GenBank files includes locus, definition, accession number, version, keyword, source, organism, reference, authors, journal, PubMed identifier references, features and so on. DDBJ. DNA Data Bank of Japan was established at NIG National Institute of Genetics, Japan. Works in collaboration with EMBL and GenBank and share data and information on Internet. Principal purpose: to improve quality of INSD, International Nucleotide Sequence Database. As public domains. Open to public and commonly shared through worldwide. Collect DNA sequence data mainly from Japanese researchers. Sequence submission tool. Sakura. Protein sequence database. Collection of sequences including translations from annotated coding regions in GenBank, RefSeq and TPA, as well as records from Swissbrot, PIR, PRF and PDB. Contains information derived from the primary sequence databases. Example: PIR, Swissbrot, Uniprot, PIR, Protein Information Resource. Integrated public bioinformatics resource to support genomic and proteomic research and scientific studies. Established by NBRF as a resource to assist researchers and customers in identification and interpretation of protein sequence information. PIR offers a variety of resources mainly oriented to assist the propagation and standardization of protein annotation. Swissbrot. High-quality annotated repository. Created by Department of Medical Biochemistry, University of Geneva and developed and maintained by European Molecular Biology Laboratory and Swiss Institute of Bioinformatics, SIB. It cross-reference with several other databases including nucleic acid and protein structure databases. Classifies data into 2 classes, core data and annotation. Uniprot. Mission: to provide scientific community with a comprehensive, high-quality and free accessible resource of protein sequence and functional information in which many entries are derived from genome sequencing projects. It comprise EBI, European Bioinformatics Institute, SIB, Swiss Institute of Bioinformatics and PIR Protein Information Resource. Comprises of 4 components, each optimized for more different uses. They are: Uniprot Knowledge Base, Uniprot Reference Clusters, Uniprot Archive, Uniprot Metagenomic and Environmental Sequences, Uniprot Knowledge Base Central Access Point for extensive curated protein information, including fusion, classification, and cross-reference. Uniprot reference clusters databases provide clustered sets of sequences from the uniprotcd and selected uniprot archive records to obtain complete coverage of sequence space at several resolutions while hiding redundant sequences. Uniprot Archive Comprehensive Repository, used to keep track of sequences and their identifiers. Uniprot Metagenomic and Environmental Sequences was a repository for metagenomic and environmental data. Protein Data Bank, PDB, repository of information about the 3D structures of large biological molecules, including proteins and nucleic acids, data derived from X-ray diffraction and NMR studies. Established in 1971 at Brookhaven National Laboratory when RCSB, Research Collaboratory for Structural Bioinformatics, became responsible for the management of PDB, it came to have an international community of users including biologists, other scientists, students, educators, media writers, illustrators, textbook authors, and general public. Funding members: RCSB PDB, USA, PDBE, Europe, and PDBJ, Japan. Keywords in Pdb file includes header, title, compound, source, author, revdar, sprayed, JRNL, secras, atom, heditum, and so on. Model Organism Database MOD, dedicated to the provision of in-depth biological data for intensively studied model organisms. Allow researchers to easily find background information on large set of genes. Plan experiments efficiently. Combine their data with existing knowledge and construct innovative hypotheses. Allow users to analyze results and interpret datasets, and the data they generate are increasingly used to describe less well-studied species. Share common approaches to collect and represent biological information wherever possible. Mods are often required for customize, capture, display, and provision data. Generate, source, and collate species specific information integrating expert knowledge with bioinformatics. Biodiversity Database, Species 2000. Federation of database organizations working closely with users, taxonomists, and sponsoring agencies. Established by International Union of Biological Sciences, IUBS. Goal to provide a uniform and validated quality index of names of all known species for use as a practical tool. The index are used to provide an electronic baseline species list for use in inventing projects worldwide. 2. For an Internet gateway to species databases worldwide. 3. A reference system for comparison between inventories. 4. A comprehensive worldwide catalog for checking the status, classification, and naming of species. Thank you all for watching. Please support us. Like, share, comment, and subscribe. We are here to help you.